Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here's what I bring to you all from the world of medicine. Another possible monkey virus spillover threat for humans. An obscure family of viruses already endemic in wild African primates and known to cause fatal Ebola-like symptoms in some monkeys is poised for spillover to humans according to new University of Colorado Boulder research published in the journal Cell. While such arteriviruses are already considered a critical threat to macaque monkeys, no human infections have been reported to date. And it is uncertain what impact the virus would have on people should it jump to species. But the authors, evoking parallels to HIV, the precursors of which originated in African monkeys, are calling for vigilance nonetheless. For the latest study, the researchers zeroed in on arteriviruses which are common among pigs and horses, but understudied among non-human primates. They look specifically at Syrian hemorrhagic fever virus or SHFV, which causes a lethal disease similar to Ebola virus disease and has caused deadly outbreaks in captive macaque galleries dating back to 1960s. The study demonstrates that a molecule or receptor called CD163 plays a key role in the biology of Syrian arteriviruses, enabling the virus to invade and cause infection of target cells. Through a series of laboratory experiments, the researchers discovered to their surprise that the virus was also remarkably adept at latching on to human version of CD163, getting inside human cells and swiftly making copies of itself. Like human immunodeficiency virus or HIV and its precursor Syrian immunodeficiency virus, SIV, Syrian arteriviruses also appear to attack immune cells, disabling key defense mechanisms and taking hold in the body long term. The authors stress that another pandemic is not imminent and the public need not be alarmed. A broad range of African monkeys already carries high viral loads of diverse arteriviruses, often without symptoms, and some species interact frequently with humans and are known to bite and scratch people. Superager brains have the superneurons. Neurons in an area of the brain responsible for memory were significantly larger in superagers compared to cognitively average peers, individuals with early stage Alzheimer's disease, and even individuals 20 to 30 years younger than superagers who are aged 80 years and older, reports a new Northwestern medicine study. These neurons did not harbor tau tangles, a signature hallmark of Alzheimer's disease. The study of superagers with exceptional memory was the first to show that these individuals carry a unique biological signature that comprises larger and healthier neurons in the entorhinal cortex that are relatively void of tau tangles. The Northwestern Superaging Research Program studies unique individuals known as superagers or 80-plus-year-olds who show exceptional memory at least as good as individuals 20 to 30 years their junior. Healthier neurons in layer 2 of the entorhinal cortex compared to the same aged peers, individuals with early stages of Alzheimer's disease, and even individuals 20 to 30 years younger. They also showed that these large layer 2 neurons were spared from the formation of tau tangles. Taken together, the findings suggest that a neuron spared from tangle formation can maintain its structural integrity. The inverse also seems to be true. Tau tangles can lead to neuronal shrinkage. Participants in the superager study donate their brains for the research. For the study, scientists examined the brains of 6 superagers, 7 cognitively average elderly individuals, 6 young individuals, and 5 individuals with early stages of Alzheimer's. Then they measured the size of neurons in layer 2 of the entorhinal cortex compared to layers 3 and 5. They also measured the presence of tau tangles in these cases. For reasons that remain unknown, cell populations in the entorhinal cortex are selectively vulnerable to tau tangle formation during normal aging and in early stages of Alzheimer's. New marker of ALS outcome identified. A study by Human Brain Project researchers has identified a new marker for predicting the clinical outcome of patients with amyotrophic lateral sclerosis or ALS through magnetoencephalography. This marker can be measured in the brain during its resting state and highlights the importance of brain flexibility for ALS patients. ALS is a neurodegenerative disease of the brain and spinal cord that causes loss of muscle control. The ability of moving, speaking, and eventually breathing is progressively impaired. There is no known cure, but treatments to improve symptoms, including magnetic stimulation, are being tested. The researchers collected magnetoencephalography data on 42 ALS patients and 42 healthy controls at the University Parthenope in Naples, whose MEG facilities have recently become part of eBrains. The new study builds on previous work by the same group, which applied the methodology to Parkinson's disease. The neuronal avalanches spread in patterns which we can monitor with whole brain scans, explains Ariana Polverino, lead author of the study. We call the collection of all unique patterns the functional repertoire, a measurement of the flexibility of the brain. The researchers focused on quantifying the functional repertoire of ALS brains even when the patient is unprompted and the brain is in a resting state. We found that a restriction of the functional repertoire corresponded to a more severe functional impairment. The more flexible the brain, the better the clinical outcome. The functional repertoire can be used as a reliable predictor of how the clinical outlook of a patient will likely evolve. Link between inflammation and cognitive problems in older breast cancer survivors reaffirmed. Scientists are still trying to understand why many breast cancer survivors experience troubling cognitive problems for years after the treatment. Inflammation is one possible culprit. A new long-term study of older breast cancer survivors, published in the Journal of Clinical Oncology and co-led by UCLA researchers, adds important evidence to that potential link. Higher levels of an inflammatory marker known as C-reactive protein were related to older breast cancer survivors reporting cognitive problems in the new study. Now, previous research has focused largely on younger women immediately after therapy, making it difficult to draw conclusions about CRP's role in long-term cognitive problems among older breast cancer survivors. In TLC, teams of researchers from around the country talked to and obtained blood samples from hundreds of breast cancer survivors and women without cancer up to 6 times over the course of 5 years. The study was motivated by hearing from survivors and advocates that cognitive problems are one of their major worries. Cognition from the perspective of each woman was evaluated through a commonly used questionnaire assessing how the women perceive their ability to remember things like names and direction, ability to concentrate, and other aspects of everyday life. The study found higher CRP levels among survivors were predictive of lower reported cognitive function among breast cancer survivors. There was no similar relationship between CRP levels and reported cognition in the women without cancer. Cognitive performance, as measured by standardized neuropsychological tests, failed to show a link between CRP and cognition. The authors say this may indicate women are more sensitive to differences in their everyday cognitive function, self-reporting changes that other tests miss. The authors said their study supports the need for research on whether interventions that can lower inflammation, including increased physical activity, better sleep, and anti-inflammatory medications, may prevent or reduce cognitive concerns in older breast cancer survivors. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.